Cool. Hi, everyone. Welcome to those who are new to Malaysian Research Insider session, and to those who have joined us last Saturday, welcome back. I'm Apple. I'm doing my PhD in molecular plant science in University of Edinburgh. This science chat is co-organized by MBIOS, 100 Scientists of Malaysia, and our media partner, Science Media Centre Malaysia. So let's welcome our scientist today, Prakrithi from Sarawak, in Monash University Malaysia, doing her PhD in cancer biology. And I met Prakrithi in University of Manchester. Was it 3 or 4 years now? Probably 4. Yeah, in a
4 years ago.
Yeah, God, that's a long time. We met at an event run by young Malaysian engineers called Picha Kucha. So over to you, Prakrithi.
Thank you. Thank you for the introduction, Apple. So I'll just start by sharing my screen and just make sure that everyone can see my screen. Yep. Cool. So good evening from Malaysia. I hope all of you are well, safe, and healthy. So today, I'd like to share with you about my journey as a scientist and how I got to where I am today. I'll also be sharing a little bit about my PhD project with all of you. I completed my secondary school education in SMK St. Mary's. At this stage in life, biology was my favorite subject. Having witnessed family members succumb to cancer, I had the urge to find a cure for cancer. So after that, I did my A-levels in Methodist College, Kuala Lumpur, where I was further inspired by my biology lecturers to pursue a degree in biomedical sciences at the University of Manchester. And some things that I really appreciate about studying in Manchester was that there was opportunity to undertake a wide array of research experiences. In the first year of my undergraduate degree, I participated in the International Genetically Engineered Machine competition. The aim of this competition was to make microorganisms perform a function that they don't usually perform. So this experience was an eye-opener for me because I learned lots of concepts and theories in my degree in uni, but I never really got a chance to apply these theories in real life, and participating in iGEM was that experience for me. It was definitely a steep learning curve because we had to perform experiments within the given time frame, and also make sure that whatever we were spending for the experiments, our travel costs and everything was within the budget that was allocated to us. And we had to manage the expectations of our supervisors, raise funds for the competition. Most importantly, we had to learn how to work as a team and achieve a common goal, which was to keep persevering despite the fact that there were multiple occasions when experiments wouldn't work for prolonged periods, and to remain motivated at all times. Throughout the duration of this project, I did ask myself this question many times. Prakrithi, is this really what you want to do for the rest of your life, like working in a lab and being in this situation? But the more I asked myself that question and the more I did research, I realized that this was one thing I really enjoyed doing, and I don't think I can imagine doing any other thing in life now. So what I did next was to gain more experience. And this time, I attended a summer school in Imperial College London. The highlight for me was being able to conduct a mini project in order to find out what activates your a particular subset of immune cells using some of the best laboratory equipments in Imperial itself. And also, I made lots of friends during that time. When I was in my final year of my degree, I was contemplating between doing an MSc and an MRes. So what happened was I went to the fair where they had organized sort of information about having the options to do further research in terms of postgraduate degrees. And when I spoke to the coordinator for the MRes course, he gave me very sound advice, and he said, well, if you're doing an MRes and you are sure that you want to do research, this would be a condensed version of a PhD with some additional taught modules. So that sort of, gave me the reason to do a a master's by research in oncology at the University of Manchester. So as you can see, this is the space that I spent the most amount of my time, the labs that I worked in. And I must say, my MRes year was very, very intense. My supervisor did warn me beforehand that he had high expectations. So in a way, it was really good for me because I got to improve my scientific writing and presentation skills. But at the same time, occasionally I would experience imposter syndrome where I doubt myself whether I actually know stuff, whether I am capable of doing research, and is this the right path for me? But luckily for me, I had colleagues who were extremely reassuring, and they would answer all of my questions patiently, no matter how many times I asked. And also, they trained me to be a scientist who learns how to troubleshoot when there are problems. And that's something that I'm really grateful for till today. So, for example, while I was working in my project was all about Fanconi anemia, which is a rare genetic disorder which causes people to be more prone to DNA damage, and also their DNA cannot repair itself. So it's like a double hit for them, and they do not live very long because they get multiple cancers, and the life expectancy for those with Fanconi anemia, it's only about 50 years. It used to be 30 long ago before there were improvements in terms of treatment. So one of the proteins that I was supposed to identify and work with just refused to appear in my Western blot experiments. And so despite trying to change a variety of things, it still didn't work. One of my colleagues has a lot of experience. Instead of using machines to detect, he actually taught me how to use films to view this protein. And that's something that a lot of people have not worked after we've had machines. So I'm so grateful for small things like that, which really helped me in a lot of ways. And one thing that I think was very important for me was all the skills that I learned through all my research experiences. Because looking back now, the things that I did in iGEM and in my summer school as well as my master's, all of them are so relevant because it's helping me during my PhD. And knowing those techniques, understanding why you are doing certain things, nothing ever goes to waste. It really helps you, and you never know which point of life it will help you in. So once I finished my master's, I did an internship at Cancer Research Malaysia, where I was part of the public outreach team. Here, I learned a lot about breast cancer in Southeast Asia and also aided in the launch of patient navigation programs for breast cancer patients. It was during this period that I received the offer to do a PhD on glioblastoma in Monash University Malaysia, and I am based at the Brain Research Institute. The title of my project is The Role of Estrogen Receptors in Modulating the Motility of Glioblastoma Through the Regulation of G protein coupled receptors. So I know a lot of you may be thinking, oh my God, this is a mouthful. Or some of you may be seeing stars. I don't understand what she's talking, but allow me to break this down for you. And hopefully by the end of the talk, you will understand at least a little bit of what I'm doing. Glioblastoma is the most common type of brain tumor that occurs in adults. Most people with glioblastoma have a very poor survival rate of 12 to 15 months, as it is a very aggressive grade of brain tumor known to invade neighboring brain regions. So it starts off small and it invades neighboring brain regions. Current treatment of glioblastoma includes surgical resection of tumor followed by chemotherapy with a drug that's called temozolomide, and radiotherapy as well. So as we saw earlier, the median survival, despite patients having gone through all of these treatments, is only 12 to 15 months. So I decided to take a closer look at the statistics of glioblastoma. And when I did, I intriguingly found that men are twice more likely to develop glioblastoma in comparison with women. So why is this so? Why is it that men get this type of cancer more than women? What is it that is protecting women per se? So there are two groups of women if we look at it closely: premenopausal women and postmenopausal women. In premenopausal women, they've got higher levels of estrogen. Postmenopausal women have lower levels of estrogen, and this therefore leads to a higher chance of developing glioblastoma. So why is this so, and what is estrogen? Estrogen is a primary female sex hormone that regulates female reproduction processes and the development of female secondary sex characteristics. Estrogen is known to bind to estrogen receptors, and it modulates the expression of G protein coupled receptors, transcription factors, and kinases. So now you may be thinking, what does this mean? Let's take the analogy of a domino. So let's imagine that estrogen here is the white figure. And when estrogen pushes the dominoes, all of the dominoes start falling. But in reality, it's not just one stack of dominoes. It may be multiple chambers of dominoes in many, many places. So if we take estrogen to be the green domino blocks, it could have its effects on G protein coupled receptors, which could be represented by the yellow domino blocks, and many other things like transcription factors and kinases, which are the orange, red, blue, and green domino blocks. So what are G protein coupled receptors? Let's look at the analogy of a post office. Let's assume that G protein coupled receptors are the post office and those who work in the post office. What happens is I am going to be receiving, let's say I work in the post office and I'm a G protein coupled receptor. I'm going to be receiving post or messages. I'm going to start calling these messages from people. Then what I do is I look at the messages that I have received, identify who I need to send those messages to, and then give it to the right postman so that they can deliver those messages to the right people. So that's exactly what G protein coupled receptors do. But let's take a look at it in real life. G protein coupled receptors are actually found on the surface of a cell, and this is what it looks like. G protein coupled receptors are those that communicate and share messages from the outside world to the inside world and make sure that the right messages reach the right people at the right time as well. So what are G protein coupled receptors going to do, and which G protein coupled receptors am I going to look at? I'm going to identify those which send me messages to tell tumor to stop growing or the tumor to start growing. So those are the G protein coupled receptors that I wanted to look at. Specifically, a family of 3 G protein coupled receptors, namely spexin, galanin, and the kisspeptin receptor. Why particularly these 3? Because spexin in particular has been shown to have tumor stopping effects in glioblastoma itself. However, not much study has been done on galanin and kisspeptin, and that's what my project will focus on. So kisspeptin receptors, what are they? They bind to kisspeptin, as is shown in this image, and they are known to suppress metastasis, which is the spread of tumor to other regions in your body. So what does my project actually look at? It's going to see how estrogen, which is the green domino block, going to have how does estrogen affect the kisspeptin receptor, and how does that influence the growth of glioblastoma? Does it stop the growth or does it promote the growth of glioblastoma? And then the other receptor that I'm going to look at is galanin receptor. Galanin receptor binds to galanin and spexin. It has a high expression in renal cancer. Now, we don't know much about its expression in glioblastoma, so my study is going to find out, does this play a role in the invasion and progression of glioblastoma? So again, my project will be looking at how estrogen influences the galanin receptor and ultimately how this affects the growth or the inhibition of growth in glioblastoma. That's what I'm going to find out. Now, how is any of this relevant, you may ask? It just looks like I'm looking at the role of receptors. Well, that's true. I am going to look at the role of receptors, and hopefully I find that either these receptors in combination or on their own either cause more tumor or stop the growth of tumor. If they are stopping the growth of tumor, I'd still like to find out how, because then I can identify potential targets that could be used as treatment for glioblastoma. So hopefully by now you understand a little bit about my project, and I hope that you enjoyed the presentation as well. Before I end, I would like to thank my supervisors, all of my lab mates, and everyone who's been extremely supportive throughout my career as a scientist. I would also like to take this opportunity to thank my family who've been extremely supportive throughout my journey. Last but not least, let me also thank God for this wonderful opportunity and MBio's 100 Scientists of Malaysia and Science Media Center for making this a reality today, for me, for being able to share my science with all of you. Thank you.
Thanks, Prakrithi. Just to let you guys know, for those who joined late, Prakrithi just started her PhD in February, so I doubt she has any data to show about her research, but thank you for the talk. I think you have a voice that really suits some kind of recording that can be played in a train station or something.
Thank you.
Right. I just wanted to ask, you know, when you mentioned spexin and galanin, so what are they? Are they hormones as well, like estrogen, or what type of molecules are they?
So spexin and galanin happen to be neuropeptides, actually. So they are found in the brain, and if we can find something that is within the brain itself, something that we don't have to introduce externally, that might be better. So that's the reason why we started looking at neuropeptides.
On that, just to let you guys know, I think men also have estrogen, right?
Yes,
not
they
just
do.
Women. Okay.
Yep. But at a much lower level.
So when you're doing these your studies, are they in women only or in men as well?
So thankfully for glioblastoma, we've got cell lines from both male and female patients. Mm-hmm. But for the start of my research, I'm just going to use male samples, and then based on the results, probably at a later stage, use female cell lines as well.
Right. Is there a reason why you're using men first?
The only reason is those cell lines are already readily available in our lab, so it's much easier to start work on those. Yeah.
Yeah, fair enough.
While waiting, yep.
Okay. Right, we have a question from Sean. Sean also did his research in cancer research Malaysia, if you know him. Mm-hmm. So he said, your hypothesis is that estrogen confers a protective effect against glioblastoma. Do men also develop glioblastoma at an older age?
Yes, they do.
They do.
So men, it also increases the chances of getting glioblastoma increases with age as well.
So do men also have higher levels of estrogen when they get older? Like, because I know women has pre and post menstrual levels of estrogen, but do men get that as well?
I think for men, it's pretty similar throughout. There's no fluctuation in the expression of estrogen. There might be slight decreases as you get older because, you know, the hormone producing ducts and all may not be working as well. So there may be a slight decrease as you age, but otherwise I think it's pretty similar level of expression.
Cool. Sorry for being sniffly, guys. Having a flu today. We've got a question from Izad. So there are some studies that mention that glioblastoma tumors are very, very aggressive post-surgical. Are there any new findings that determine any genetic or biological pathway that might be the cause of this behavior, this aggressive behavior?
As of now, there's no specific cause per se. It just happens for them. So there are no causes per se. Some say that maybe exposure to radiation at an early stage in life could probably cause it, but otherwise there's no real link to genetic factors.
Oh, right. So people have done research on the genome and they found no association with any sort of polymorphisms or
No, as
mutations.
of now, no.
Oh, interesting. Okay, there's a question from Jake. I'm not really sure what GPCRs are. Do you know? G coupled proteins.
G protein coupled receptors.
Oh, messed it up. Okay, so he's asking whether all these G receptors are expressed solely in the brain.
No, that's not true. G protein coupled receptors are actually expressed everywhere, but the ones that I'm looking at are expressed more in the brain.
Okay, that's cool. So Tom asked, what model will you look at for galanin receptor expression?
So I would start off looking through cell lines, glioblastoma cell lines, of course. And once I complete that, hopefully in the final year of my project, I would be able to also do studies on animal models.
Okay. Which probably rat.
Probably mouse or rat. Yeah.
That's cool. If you could talk more about your iGEM research, that would be great because I found that really, really interesting. What do you do in iGEM?
So for iGEM, I think it was a lot of genetically engineering microorganisms. So during the project, what we wanted to do was we had to identify a problem, and the problem was there was a lot of drinking issues in Manchester itself. So how do we sort of improve on the testing methods that are already out there? Currently, we've got things like breathalyzers and all, but they are bulky and expensive if you want to get a good one. So then we looked up literature and found out that your blood and sweat alcohol levels are linear. So that means that you don't necessarily have to do blood testing for alcohol. You could potentially also do sweat testing for alcohol, which is where we got the basis of our idea. And if you can use sweat, which is a non-invasive method to detect how much alcohol you've consumed, wouldn't it be good if we could have something like a patch or like a wet wipes that would, you can just wipe your sweat off and it would immediately change color to indicate, okay, this is how much you've drunk and this is your alcohol intoxication level now. So we use the traffic light system with red indicating extremely high levels of alcohol and green indicating very low levels of alcohol. So what we did was genetically modify E. coli, which is a bacteria, and introduce genes from Aspergillus, which is a plant, and that has a mechanism of detection of alcohol. So we introduced the genes from Aspergillus into E. coli, and we wanted to make them change colors. So we got to the stage where we had blue and pink E. coli, but we hadn't actually done the other colors and all. So it was more of a proof of concept. We got the bacteria to change color and sort of react to different levels of alcohol. And we also did some modeling to help us. So yeah, that's about it.
Yeah, that's pretty cool. So just to correct you, Aspergillus, I think is a fungus, not really a plant.
Oh, sorry. Yep, my bad.
No, that's fine. So back to your research in glioblastoma. So Jake has a question.
Yep.
So with the ligands that bind to the G proteins, I think spexin and galanin, are they
Mhm.
circulatory in the blood? And can they be used for biomarkers for glioblastoma?
So spexin and galanin are actually found in several other locations in your body as well. So they are circulatory, but more expressed in the brain. So yeah, potentially spexin I don't think could be used as a biomarker because it's highly expressed in renal cancer and not in any other cancer types. So probably we're not sure about galanin. No research has been done. So something that I could probably answer in a few years' time if it's a possibility to be used as a biomarker.
Yeah, cool, cool. So Jerry asks, can you just name a few research techniques that you would use in your PhD studies for glioblastoma?
Sure. So the technique that I'm currently doing is PCR, and after this I would be doing some Western plots as well, cell culture, and also probably maybe down the line some flow cytometry possibly.
Cool. Do you still want to continue doing research in cancer? I know this is a quite early on, but maybe after your PhD, what do you aim to achieve? Do you want to become a supervisor, a principal investigator, or?
Yeah, so I definitely want to continue doing cancer research. And I think probably down the line, having my own lab and working on lots of cancer research. I think one thing that's really intriguing, particularly now with all the advances in cancer treatment, is how the immune system is involved in cancer as well, to
Mhm.
see how the crosstalk and if possibly we can use our own immune system to fight cancer. So that's something that I'm quite interested in as well.
Yeah, that's quite interesting. Do you know of any research that's doing that, trying to use the immune system to fight cancer?
Yep. So there are quite a few labs all around who are working on that at the minute. So yeah.
So how do you trigger the immune system to do so? Do you take some kind of drugs or?
So one thing that you can do is actually extract immune cells from the patient, and you also extract the tumor cells from the patient. So then what happens is you identify what proteins are expressed on the tumor and also identify what proteins are expressed on your immune cells. And then if there are particular markers which can help your immune cells further identify the tumor, we add those to the immune cells. So it's sort of re-engineering the immune cells and then injecting back into the patient so that it recognizes the tumor. Because what happens is most often tumor cells adapt and they appear as host cells. So one way of making sure that your immune system actually recognizes is to add these additional proteins onto the surface of your immune cells.
Yeah, and then yeah, CAR T immunotherapy, like what
Yep,
Liang said.
exactly.
Yep. Um, just coming back to estrogen again, because we had a Research Insider session, I think 12 or 13, where Sean talked about heterogeneity in cancers itself. So for those who
Yep.
do not know what heterogeneity means, that means that in a tumor itself, each cells have their own individual expression profiles in terms of um, gene expression. So how are you gonna um, include that in your research?
So that is one aspect that a lot of people are dealing with because technically we have drugs out there, but it's not a one size fit all kind of treatment. Everybody, if two people have the same cancer, the cancer is not going to be the same. It wouldn't react the same way to treatments and things like that. So in an ideal world, we would have specialized treatments for each patient. We would do genetic screening, identify what are the genes present, how can we best treat this patient, what are the combinations of therapies and treatments that we can give. So in an ideal world, I'd like to believe that every patient would have, how we have phone number catalogs, but instead we'd have actually gene profile catalogs and then say, okay, this combination works best for this patient.
Yeah.
So
Yeah,
hopefully
that's
someday
true.
that happens.
Hopefully. So yeah, again, back to your iGEM experience. There's a question by Jake. He asked, what's the most commonly used technique during your iGEM? Is it cloning?
Yep, lots and lots of cloning, and there was one period where it wasn't really working. It wasn't just us, we had our advisors try as well, and somehow none of them could actually figure out why it wasn't working. And then later on we found out that it was actually an issue with one of the parts that we were using, but by the time we figured out, it was a bit too late. So we did lots of cloning, PCR, gel electrophoresis. Yeah, those were mainly the three techniques that we used.
Yeah, so there's a general question again from Jake from your experience studying overseas. So what do you feel was most useful from your mentors?
I think each of my mentors taught me different things. So I'd like to start off with my mentor from my final year project. He is the most optimistic person I've met in my life, and his optimism actually rubs off you. The other thing is he's extremely organized as well. So in terms of doing my final year project, there was a beautifully set plan, and I think it also set some expectations for the next research projects because I was so used to that type of work. And my master's project after that was the complete opposite. Um, so we didn't really have a plan. You go into a lab, and if they are working on something exciting, you join them and you help them with their project because you've only got about 9 months of research. And, you can't be possibly doing a lot and start an entire project on your own. It's much easier if you fit into the grand scheme of things that are happening in the lab. So with that project, I really learned how to be very resilient. There were weeks of trying to detect my proteins and things wouldn't work sometimes, or you would have to repeat because your results do not match and how you cannot publish such data then. So to be really resilient, to work, and also for my internship experience, I had a supervisor who was very, very strong about how we should have a work-life balance. She was saying this because when she started her career, she did not have any form of work-life balance. It was all work and no play. And so she would actually ask all of us to make sure that we stick to the work hours and not take any work back home. And at 4:30 every evening, she would pack up her bags. She'd be the first one, and she'd say, "Okay, it's time to go home now. The school bus is ready." Um, so yeah, different things from different supervisors, and most importantly, I think through all these experiences, I really forged lots of lasting friendships. And there's always someone to discuss science with at any stage in life. So yeah.
Yeah, that's true. I was just gonna say when you mentioned that your supervisor said that I have high expectations from you, like that's a bit
Yep.
that's quite rare for a for a supervisor to say to you.
That's
But
true. His
yeah.
actual words were, "I'm a very difficult person, and many people don't like that." And I understand why people didn't like that. He had expectations in the sense that as a master's student, he expected me to write like a PhD student.
Mm.
But at the same time, he also understood that he's been working in this particular field for 16 years, and he's had 16 years of knowledge, whereas I've only got probably 6 months of knowledge at that point when I started writing. So then he realized that, oh, yes, I forgot about this. He sat me down and actually went through the entire thing. And he was like, "This is why I'm doing this. This is why I'm doing this. And this." And he just explained all of it really clearly and well to me. So yeah, I think I've been quite lucky in terms of having great supervisors throughout.
Yeah, yeah. That was pretty. I would take that. I would be pretty shocked if someone says that to me.
Yeah, so the only thing that I could say as a response was okay. And then he questioned me and he was like, "That's your response?" And I was like,
Oh no.
yes, I don't know what else to say. So I said, "Well, I'm taking this as a challenge." And I think at the end of it, he did say, after seeing my final presentation, that he's really proud of seeing how I've progressed. And another thing that he would do, he would actually ask all of us in the lab questions that we didn't know the answer to, but a lot of people didn't like that. The
Hmm.
reason behind him doing that was to make sure that, he would even go to the extent of looking at your posture, whether you shrink when you're scared and worried,
Oh.
or whether you maintain your posture and you say, "I don't know the answer to that, but I'm happy to look it up." So I think it was really great working with my master's supervisor in that sense.
Yeah. Yeah. That's It teaches you how to be tolerant, I guess.
Yeah, and how to deal with people. I think
Yeah.
one other thing is, like, initially while I was doing um, looking out for masters and PhDs and all, I think I was more of looking at the research topic. But one thing that I actually learned throughout my journey is that you also need to see if you are a fit in the lab with your supervisors, the people who you're working with, and my current supervisor. He really doesn't care about your grades, doesn't care if you've got publications whatsoever. But for him, he needs to see whether you fit as a person in his lab. And I think that's also another important aspect, because you spend so much of time in the lab and you really need to get along with those people who you're working with.
Yeah, following on to that, like Jade asked, like how would you advise new research students to figure out whether you fit into the lab group?
Okay. So one thing is actually looking at previous students who've worked in the lab, or research assistants or postdocs, whoever you get a hold of. Ask them about the lab, ask them about their experience, and also other PIs who've worked with the lab that you are going to work in, see if they've got collaboration, and yeah, just speak and ask people around how it is. Visit the
Yeah.
place, get a feel of it, if your vibes match.
Yeah, actually, for my PhD, when I got accepted after the interview, like that was the first thing they asked me to do. I went to Edinburgh. They asked me to assess whether I fit into the lab, and I could
Yep.
reject the funding if I don't feel like I like the supervisor or I like the lab environment. So,
Mm-hmm.
for those in the audience, having the right supervisor and the right colleagues do matter a lot
Yep.
on your PhD success. So I think that's thank you Prakrithi for coming to speak to us on Saturday night. Yeah, and thank you to the
Yep.
audience who joined us today. So our scientist next week will be Lukman Fatah. He's doing his PhD in genetics in Kyoto University in Japan. So he looks at the role of jumping genes, aka mobile elements, in the human genome. So for more updates regarding future research inside the series, like our Facebook page and bios and 100 Scientists of Malaysia. And see you guys next Saturday. Bye.